First, thanks a lot for the invitation. It's a fantastic city. I'm slowly figuring out how to decipher the alphabet. So hopefully I'm doing a decent job here. So the reason why we are here mostly is because of the high throughput sequencing technologies. Since 2008, they have been changing the world by providing a much higher throughput than Sanger sequencing and lowering the cost significantly. And lowering the size too, hopefully. They obviously come with caveats, but they're not that important. Hopefully, they will help us solve some of the big problems in genomics. First of all, we'd like to be able to compare genomes, see huge differences between them, especially structural differences between them. And hopefully, the technologies will help us solve this problem. But together with these capabilities, new problems emerge. For example, how do we handle the exponential growth in data size? And although these two big problems seem far apart, there are some links between them. And let's try to explore what these links may be. First, I'm going to focus on the first problem. How do we compare genomes, especially with respect to structural changes? One of the capabilities of sequencing technologies is the ability to pair and sequence. So especially if you're given a DNA fragment of known length, we have the capability to sequence both a prefix and a suffix of the fragment, provided that the fragment length is coming from a tight normal distribution. We can use this to figure out structural changes between a donor genome and perhaps a reference. So idea one, with respect to genomic structural variation discovery, is to find out the differences between a donor genome sequenced through one of these technologies and the reference genome. Maybe you'll do the same with a second donor genome and then reconcile the differences. To be able to do that, perhaps we should first focus on discordantly mapping reads to the reference genome. And by discordantly mapping reads, I mean those whose mappings change the distance between the two ends, or there's a change in orientation. An example is here. Suppose that I have this read coming from the donor genome. If they map to the reference genome such that the distance between them increases, clearly there's a segment here in the reference genome that is missing from the donor genome. So there's a deletion in the donor genome with respect to the reference. You can come up with similar rules for novel sequence insertion, inversion, and so on. However, when we are saying all this, we are making some implicit assumptions. In particular, we are assuming that the mapping is unique. So obviously, if the read maps uniquely to a single loci, our job is easy. If it's not the case, we have to think about it. We also assume no overlaps exist between structural variants. So these block operations are well separated, which is not correct. And finally, we assume that if we know the differences between donor A and the reference, donor B and the reference, then putting them together, we'll be able to figure out the differences between A and B. And this is not always correct. So let's focus on the first two major issues, major assumptions, and see what we have been able to do and what we haven't been able to do. I was told that I have to come up with some open problems, and there will be some open problems during the talk. So some of you may have heard about the mapping tools that we have developed over the years. Mr. and Mrs. Fast now have the new versions. There's nothing magical about them. Algorithmically, they're very simple. It's a seed and extend type paradigm. I like the fact that they're optimally cache oblivious, but there's nothing fancy about that either. However, they provide all mapping loci under certain constraints for all reads in the genome. So you take a fragment, You map all the left ends, all the right ends, and then try to figure out whether there's a concordant mapping. Concordant is when the distance and the orientations come as you expect. If all mappings are discordant, then each such read implies a structural difference between the reference and the donor. And if there are multiple mapping loci, then we have a program called Variation Hunter to resolve the mapping ambiguity. And I'm going to first focus on the maximum parsimony-based structural variation discovery through Variation Hunter today. Here are some old figures, a comparison of Mr. and Mrs. Fast with respect to some of the well-known mappers. These are old figures. The new figures still compare quite well, especially if your goal is to do structural variation discovery. And for that, you have to find all mapping loci. Anyway, let's focus on Variation Hunter. So each discordant pair then mapping obviously implies a structural variant because they don't have a concordant mapping. If you'd like to do maximum parsimony-based structural variation discovery, we'd like to cluster these read mappings together as much as possible to explain with the smallest number of structural variants the maximum number of reads that are discordant. For this, you can obtain maximal clusters, those read mappings that can be clustered together so that they imply a single structural variation. And then go through each one of those clusters through a very simple combinatorial optimization framework. If you think about it, this turns out to be identical to the set cover problem because the sets are these clusters. In each cluster, there are multiple reads that can be mapped to. And you'd like to figure out the minimum number of sets that cover all these reads. And you apply, obviously, the famous greedy algorithm that provides a logarithmic approximation. And that turns out to be a tight bound. So this is not magical. The key issue occurs when structural variants overlap. First of all, even under the assumption that the structural variants are distinct and distant, our formulation so far does nothing to avoid this kind of a scenario. Suppose that one cluster implies a deletion here, another cluster implies a deletion here. Under the assumption that we are dealing with a haploid organism, this scenario is invalid. We should avoid one if we pick the other. So we extended our formulation so that when we are going in an iterative fashion, greedily, and pick up the cluster that has higher support than that with a lower support, we focus on just this, and then we have to prohibit each such cluster. As soon as you have these conflict graphs embedded in the problem, there's no theoretical guarantee. That's pretty unfortunate. So I'm not sure if the problem is clear. We have, again, a set cover formulation. We are going through the sets as the greedy set cover algorithm implies. However, during the iterations, if we come up with a set that we have to pick, whose implied structural variation conflicts with an already committed structural variation, then we avoid it. As soon as you have this heuristic, you don't have any theoretical guarantee. So what we know is, as soon as there's an issue of conflict resolution, the problem becomes not only NP hard, as usual, but we know that it's NP hard even to approximate it with an n to the power 1 minus epsilon factor for any epsilon. So in fact, for the general problem, for arbitrary conflicts, there's no theoretical guarantee possible. However, as I'm going to show, there may be opportunities for certain cases in which, say, there are at most 2 conflicts possible, 2 structural variations overlapping. For that specific case, limited number of overlapping SVs, we don't know what can be achieved. Second issue is, so far, we don't know whether certain structural variants can overlap or not. And yes, 2 deletions cannot overlap, but 2 inversions can. And we haven't done anything about that. This is a big open problem. How can you come up with a formulation that allows overlapping structural variants? So I think those are 2 things that I'd like to pose in this context. Okay, as I mentioned, the implicit assumptions that we were making, or other people are making in, say, the 10,000 Genomes project, is that, number 1, there are unique mappings. Variation until expands that, doesn't have that assumption anymore. And then there are no overlaps between structural variants. Variation until totally avoids that, and this is one big open problem. But the bigger problem, I think, is when we say, if you figure out the difference between A and the reference, B and the reference, we can combine them together to find their differences. So, yeah, to summarize, we have those reads, we have their mappings, we establish clusters, and go through the clusters to figure out which of these, the smallest number of clusters that can explain all the reads. So this general strategy has to change, especially when we are dealing with donor genomes that are very close to each other. So you have a healthy tissue versus the cancer tissue from the same individual. You have time series data. You're comparing a child with the parents. You cannot use the old paradigm because those 2 tissues are very, very similar in comparison to the reference genome. So I was looking at the flight times from Istanbul to Moscow and Istanbul to St. Petersburg. Roughly, they're both 3 hours. So I can deduce that Moscow is St. Petersburg. Then when I arrived in Moscow, I figured that it's not exactly the case. You still have to fly for an hour and a half. And the same situation exists here. The reference genome cannot be used as an intermediary exactly if the 2 genomes are very similar. So we did the following experiment. We looked at the famed Yoruban family. This is one of the first trios that have been sequenced, and the data is publicly available. We looked at the child in comparison to the father and the mother, and we wanted to find out novel ALU insertions. And when variation hunter is applied to this data set, we discovered 410 novel ALU inserts in the child that are not present in mother or father. And this is very surprising because you would expect 1 new ALU insertions in every 20 births. So this is really, really wrong. So we came up with the following updated model. Here, we'd like to analyze these clusters formed by both the child mappings and the mother and father mappings simultaneously. The program that does this is called Common Law, and we presented the general idea a bit more than a year ago. And it goes as follows. We have a set of those discordant pairs and reads from donor genome A and donor genome B. And then we try to cluster them as much as possible together, such that we focus on those clusters that include reads from both donor genome A and donor genome B. So we have to change the general approach a bit. And in fact, from a theoretical point of view, you can come up with an optimization problem where the goal is minimizing a weighted sum of structural differences between A and reference, B and reference, and A and B all together. So I'm not going to get into the theoretical details, but in general, the cost will be a sum of all those differences between pairs of genomes multiplied with the appropriate weight, where the weight determines the genomic distance between the donor and the reference genomes. So under this formulation, if we have the black genome and the blue genome, the mappings can be coordinated, clustered in a way that we pick the clusters that involve members from both genomes first. And those that are including members of only one genome are picked the last. That simple greedy heuristic seems to do well. In fact, it provides an approximation factor that matches a lower bound. Maybe you care about this, maybe you don't, but it's refreshing to know. It's kind of nice to know that what you do in practice also fits theory. So after applying the simple greedy method, we noticed that, at least in this particular example, the number of ALU inserts in the child that cannot be found in the parents dropped down to 20. This is good, but still not good, because all those 20 turn out to be not true. So we worked on some additional heuristics and added the conflict resolution capability. And as soon as you do that, the number of unique ALU inserts turn out to be 0, as expected. Well, this is not good enough. I mean, yeah, this may be the right number, but how about the other numbers? So we went through figuring out novel deletions in the mother, the father, and the child. And we went through some of the known Fosmid-validated deletions, and we tried to find out how many of them we can find. And it seems like the heuristic with conflict resolution seems to do much better than certainly the independent discovery method, and also the first heuristic that I mentioned. Okay, so as you can see, Variation Hunter sort of takes care of unique mapping issue, does not take care of overlapping structural variants. And common law seems to take care of the reference genome issue, but there are still certain issues. You're still biased by the use of the reference genome. So alternatively, one can assemble donor genomes independently and then compare the assembled genomes without the reference. And we see this idea popping up everywhere. However, it has some major issues. As we have seen yesterday, de novo assembly problem is far from resolved, especially at human scale. And comparison of assembled genomes problem, again, as seen yesterday, is very difficult. It's generally unresolved, especially when structural variants overlap. There's an emerging idea, starting with the program Cortex, of assembling differences between two donor genomes. So on a colored de Broglie graph, you can find out uniquely colored regions to find unique inserts in one donor genome versus the other, which seems to work to a certain degree. But I haven't seen a comparison of Cortex or other methods with mapping-based variant detectors in a meaningful manner. Although the goal is to incorporate Cortex in the Pilot 2 study of 1,000 genomes, I haven't seen the solid numbers yet. So I can imagine that differential assemblers will be able to solve some problems, but certainly not all. So let's focus on, under the assumption that we can indeed assemble genomes de novo, what happens when you want to really compare two genomes. For that, we know the block edit distance seemed to capture many of the edits that we have been seeing in the last 2 days. So block edit distance is defined as the minimum number of single character edits, as well as block or segmental edits, such as inversions, moves, copies. And to make this a distance, symmetric distance on copies of arbitrary length. So block edit distance was shown to be NP-hard more than 15 years ago. But we know that it can be approximated with an almost logarithmic factor in polynomial time. So there are limited versions of block edit distance, such as the edit distance with moose problem. It's NP-hard as well, but it can be approximated much better. There's a constant approximation, and perhaps this can be improved. So, okay. What is the link between variant discovery and handling exponential increase in sequence data? So let me go through one game, a two-party communication problem in which Alice and Bob have genome sequences A and B respectively, and they'd like to exchange these two sequences. In a typical scenario, A and B differ by, say, a few million single character edits and a few tens of thousands of block edits. Obviously, it's possible to embed A and B to vectors, say, capital A and capital B, through a paper that I was involved with more than 12 years ago now, so that the block edit distance between A and B will be upper bounded by the L1 distance by these embeddings of A and B. And this will be, again, upper bounded by the block edit distance times log of the lengths of A and B. So basically, this is a low distortion embedding that takes strings or sequences under block edits to vectors under L1 distance. This becomes a vector comparison problem. And the interesting thing is, once you have an embedding like that, you can use communication protocols by Orlitsky et al. Developed in the 90s to be able to exchange, to be able to say, have Alice send Bob what she has by not communicating the whole string, but by effectively encoding the differences between what she has and what Bob has without knowing what Bob has. Okay, so perhaps some practical version of this should solve some of the problems that a future index of global genome sequences should have. Currently, I don't know what you guys do, but if I want to send somebody my donor genome sequences, I put them in a hard disk, send it by UPS. This is not sustainable. We have to do something to improve the communication. And also, storage is an issue. The growth in sequence data is outpacing Moore's law, as everybody says. So we have to do something to improve compression. Perhaps, as long as we keep the genomes as a collection of reads with all the quality scores, names, and etc., we are stuck to limited compression methods. As we start to keep things in assembled form, maybe things will improve. But even when you're dealing with high throughput sequence data, perhaps the quality scores will diminish in time, but at least we'd like to keep the reads as they are. Maybe things can be improved over what most people use, like Lempel-Ziv-based techniques. So as far as I know, most sequencing centers just use gzip to compress all their data. They store it that way, they communicate it that way. So we start to think in these lines and try to figure out whether there's a way to improve the performance of gzip on high throughput sequence genomes. So the first thing that comes to our mind is to improve the locality of reference. If we don't do anything, so this line shows the gzip performance on 5 million reads from this Pseudomonas genome. We are dealing with 51 base pair reads. The original file size turns out to be 250 megabytes, and gzip compresses it to 65 megabytes. If you do something very simple, take these reads and lexicographically sort them, suddenly you get 26 megabytes. So that's not very surprising, but if you know how gzip works, gzip basically tries to find similar substrings. In fact, it replaces similarities in substrings by pointers to, say, earlier occurrences of these substrings. And the more you improve the locality of reference, the better compression you get. A second idea is using mapping. If you take all these reads, find unique mapping locations to each one of these reads, suddenly the compression improves. And from 26 to 20, it may not look that impressive, but still, that's almost 20% of an improvement. And in human genomes, the improvement is much bigger. The reason why I don't report it is, when you're dealing with human genome scale compression, mapping takes forever, as you all know. Even at this level, if you look at pre-processing time, obviously for gzip it's 0. Lexicographic sorting is less than the time required for gzip, but mapping is much more than what gzip requires. So we cannot afford this, especially in human scale. So can we do something to replace mapping, provided that it's the computational bottleneck? So if you remember, mapping is typically based on seed and extend type algorithms. If you just do seeding, then extension. So we do the first stage of mapping, but not the second stage. Maybe that's good enough. Maybe we don't actually have to find the mapping loci of the reads. So the seeding, identifying consistent seeds from reads so that they can be clustered together, and then compressing each one of the clusters independently may provide results similar to mapping. Of course, this is easier said than done because we are not, it's not very clear how to pick up the smallest number of seeds. Maybe I should tell the problem very clearly. So suppose that I'm given a complete reference genome sequence. It's of size n. And I'd like to pick up the smallest number of seed strings of size w, such that any read of size m includes at least one of these seed strings. I don't know if this problem makes sense to you, but maybe I'd like to make it, maybe I should make it a little clearer. I'd like to pick up the smallest number of strings of size w, such that every read that can come from this genome of size m will include these seeds. This is an unresolved problem. And if you have any ideas, I'd like to hear. I'm sorry?
No, I said that the pre-type error is sometimes different.
Yeah, that's a bigger issue. In fact, I don't want to do it for the reference genome. I'd like to do it for the donor genome. In fact, just storing the reference genome, processing it, is a hassle. A better problem is the following. Is there a way that I can come up with string kernels, a smallest number of strings of user-defined length C, which can be concatenated to construct any string, so that I can use all the string kernels from these reads, pick up one in a consistent fashion, so that those strings that are represented by the same kernel are clustered together, and then I can compress them together. And I don't know how to solve this either. Instead, I do something from Cormode et al. I identify what we called core strings. I'm not going to get into this. It's a very cumbersome, difficult construction. The good thing is it's universal. It's not for a particular genome sequence. It's universal for a given alphabet and a given value of C, provided that I know my read length. I do it once, and I embed it in the data compression algorithm that I'll use. And they provide a good enough approximation. And in fact, our program called Scales, based on these cores, pretty much emulates what mapping gives us. So pre-processing time, identical to lexicographic sorting. You cannot do better than that. The gzip time is comparable to that with mapping. The better compression you get, the less time you spend on compression anyway. And the file size, the eventual file size, is pretty much identical to what mapping-based gzip achieves. So I get the best of both worlds. And I can imagine, so this is a very good example. In human genome, the results are not as impressive. If I have a good way of establishing these string kernels or solve the first problem, I'll get, I'm sure, a much better result. Okay. Now I'm wondering how much time I have.
10 minutes.
Okay. So before coming here, I actually wanted to talk about this. Maybe it's of more interest to much of you, but then I wanted to say something algorithmic. However, this is what I do to earn a living. So much of my attention these days goes to the analysis of RNA-seq data, and if possible, matched WGSS data to find aberrations in cancer, genomic and transcriptomic aberrations in cancer. Some of our recent work focused on fusion genes caused by somatic rearrangements, which should confer a competitive advantage to tumor cells. You know that they are used as biomarkers and potential drug targets. We have done a comparative study using both RNA-seq and matched WGSS, but each one obviously has its advantages. I'm not going to get into that. I'm going to focus on just some results. So by the use of RNA-seq to prioritize aberrations, express aberrations, and the genome data to classify them, we were able to obtain fusions that have been undetected in prostate cancer. So the biggest challenges that we face are figuring out whether a particular structural variation in the genome corresponds to an aberration, a fusion in this case, in RNA-seq. So this is done still in an ad hoc way, and I'm not very proud of what we do. In a way, it is an open problem. It hasn't been totally mathematically defined. But once it's done, the goal is to find, again, in a maximum parsimony manner, the smallest number of fusions that can explain all the read data. So let me depict the problem through this figure. You have the whole genome shotgun sequencing reads. You have the RNA-seq reads. You find rearrangement breakpoints. You have fusion transcripts. Then you have to find which of these correspond to which of these. And then you have to pick up the smallest number of breakpoints and the fusion transcripts that correspond to each other, which would explain all these reads here and all these reads there. So I'm not going to get into the depths of this combinatorial optimization problem. The main message here, I guess, is to corroborate these two types of events, we need better ways, better mathematical definitions. Let me see. Okay, so that was part of what we did. This is from ISMB last year. In RECOMB this year, we also explored complex balanced rearrangements. So a balanced rearrangement involves multiple chromosomes and fusions among these chromosomes, which probably occur simultaneously. So a fusion between, say, a gene here from chromosome C with a gene here in chromosome A, followed by a fusion here with chromosome B, and again followed by another fusion, can be observed quite frequently. And to be able to find out these corroborated fusion events, we had to come up with a combinatorial optimization problem again. Note that many of these cases are not expressed. Although we know that there is a fusion, say, between B and C involving regions B1 and B2, it may be possible that they are not expressed at all. So you have to somehow use both genome data and RNA-seq data to be able to find the cycle and the single event that seems to explain what's going on. So in addition to balanced rearrangements, there are unbalanced rearrangements as well. And for that, you have a shortest path formulation. Many of these are due to, we predict chromotripsis, as you have heard before. And to be able to detect these events, we came up with Enfuse, the complex rearrangement discovery tool that just appeared in Genome Research last month. So here the idea is to identify complex rearrangements. For this, we do some targeted search based on fusion transcripts. And then we find the breakpoints in the genome sequence data. And then we find shortest paths in a bipartite graph that we define to find the simplest explanation for these coordinated events. So maybe I should say just a few words about what we were able to figure out. In the C42 prostate cancer cell line, and a particular tumor sample coded 963, we were able to observe several novel fusions. In particular, in C42, we predicted 11 breakpoints for 9 fusions and validated all of them. We also find out some chimeric transcripts, those fusions that are expressed, but there's no genomic signature for them. This is an old picture, but it just gives some ideas about what is recurrent and what's not. Interestingly enough, none of the genomic breakpoints were recurrent, but many of the read-throughs and chimeric transcripts were recurrent in many of the samples that we look at, prostate cancer samples looked at. And the control wouldn't have some of these transcripts. And in addition, let me go through some of these figures. We were able to find out some complex balanced rearrangements in 963. This is one example in which the oncogene Mike was involved. As you can see, this is an example in which 4 fusions simultaneously occur, and 2 of them were expressed. More interesting pictures involve not a single breakpoint, but multiple breakpoints per loci. Again, this is a case in which 3 chromosomes were involved, but the transcript products involve pieces from each one of the 3 chromosomes. And so on and so forth. There are quite a few complex rearrangement cases that we have been able to observe. So the big open problem, which is not algorithmic in this particular case, is where's the evidence that these things occur simultaneously? So what is there a null hypothesis that one can develop? And I see evidence for these events happening. Yeah, it makes sense that they occur all together. But how do we quantify? How do we say that there's this likelihood that they occur together, and thus there must be a mechanism underlying these complex rearrangements? So with that, I conclude the talk. I'd like to thank my students, my colleagues, especially Evan Eichler, with whom I did most of these structural variation discovery studies, and with Colin Collins, with whom I've been doing all the work on prostate genomes and transcriptomes, and the funding agencies.
You seem to be critical of cortex. Can you elaborate on?
I'm not critical of cortex. I'm just, and we came up with the same idea. In fact, I remember writing you an email about that before there was even any rumor about that. However, I don't see how they can. Novel inserts, I understand. Novel insert in one against the other, I can understand how they find it. But how are they going to find copy number variation? When copy number variation overlaps with, say, an inversion, how are they going to find any of those complex events? I don't see. I don't think that it's a solution to all the problems that they think they'll solve.
But what about using it for comparison of complete?
Yeah, so I can imagine that they'll be able to find a novel insert in genome 1 which doesn't occur in genome 2, and vice versa. But copy number variation, they can't do much.
But as soon as the genomes are known, then the copy variation is not.
What do you mean? Why shouldn't it be a problem? So if my goal is to identify novel inserts, I can do it through mapping anyway.
I'm asking through the assumptions that when we compare complete genomes.
Right? So I don't understand the question.
Anyway, so I have two different questions.
Okay.
The first one is when you were saying about the difficulty in comparing was the overlapping between social variations.
Mm-hmm.
What do you mean by overlapping social
Right.
variations?
So we observed that, I have to say, Variation Hunter and similar programs, and after we published Variation Hunter, Hydra was published, and there's GASV and a number of others. One of the key things that we missed was reliance on a single type of signature. It's the mapping and the discordance. However, then after we looked at the depth of coverage, we noticed that some of the events that we thought we discovered are just wrong, because a novel ALU insertion was followed by, say, an inversion. So another example is we have seen evidence for 2 inversions simultaneously happening. We have no ways of detecting these. So a sequence is inverted, then a portion of it is inverted again. And those give conflicting signals according to this formulation. And although one can sit down and take the hammer and just carve something, which will be very, very complex, a huge case analysis, I don't like it. There must be a universal solution. It's not good enough.
Second one. When you compare two genomes, you want in one slide you say you wanted to find the breakpoint value.
Mm-hmm.
And what next?
Let me see.
Yeah.
Right here.
Just after this. Yes.
Okay. So in the breakpoint graph, it becomes it's a little more sophisticated than a standard breakpoint graph. In the complex balance rearrangement case, we look for a cycle in the digraph that we have, and the cycle. So this is a bipartite graph in which you have. I have to get into some depth, maybe after the talk. But the key issue becomes coverage of these breakpoints with the smallest weighted cycles. So we are looking for a cycle that seems to make sense. We are looking for the smallest number of fusions that can simultaneously happen to cover all the fusions that we analyzed.
There's no repeats here.
There are repeats. There are repeats. In fact, each read can be multiply mapped.
Then the definition of alternating cycle is not defined.
Why is that? We have to really go through this. I mean, this is a 60-page, nasty paper. I'm impressed that Genome Research eventually published this, but there are many details involved. We make some simplifying assumptions over the processing, but it has multiple mappings per each read.
So I have a question about genome compression.
Yeah.
Is it necessary to always transfer all data? Perhaps one could design some kind of queries for some tasks.
Yeah. I don't think it's necessary, yeah. I don't think it's necessary. It's just I try to highlight the fact that what we are doing, the current practice seems to be excessive, right? Just not being able to communicate genomes through the internet is just weird.
I have a related question. What do you think? What's a good trade-off between storing all thread quality reads and not storing it at all?
So we did a study. We did a lossy compression of the quality scores. I don't have any results here. We also use arithmetic coding. So it turns out that by simply discretizing the quality scores in any way you want, simple quantification and discretization, right?
1 to 5?
So more like 1 to 10. You don't lose mapping accuracy. So the same reads, if you look at, say, bow tie, okay, that's bow tie 2. Look at the best mapping loci. 99.9% of the reads map to the same loci, even after the loss of quality scores. So either the quality scores are not useful, that important, or the discretization works. So why don't we agree with lossy compress the quality scores, or totally getting rid of them? Doesn't seem to hurt much.
The problem is also that if you look at BAM files today, about 40% of the data is identifiers. So
Right.
unless you come up with a more, less stupid way of making identifiers,
Right.
and only about 10 to 12% is actual sequence data. So even if you achieve 10 times compression, it's not going to achieve the compression.
True.
Yeah.
I have a question about overlapping structural
Yeah.
variations. And can you say, or somebody say, how much open they occur, or how many structural variations overlap, and how many variations do not?
Yeah, that I don't know. This is a very good question because there's no way to figure them out. Here's what I know. We looked at Fosmid sequence regions, and there are some obvious examples, especially near ALUs. There are obvious deletions, for example. Next to an ALU insert, there will be a deletion. It's obvious because of depth of coverage or in inversion. And we know that we missed them. So I'll just come up with a number. Because when I say false negative rate, it's not a real false negative rate. I'm sure that we are missing even more than this. But our false negative rate among the events that we know are happening stands around 10%, maybe a bit more than 10%. So some of them must be due to overlapping SV events. I don't know if it's an answer.
So back to cooperation.
Yeah.
There's been an ongoing discussion for some time now about whether to keep raw sequences or image data. So because maybe there could be better base calling programs.
Yeah.
So in the case of image data, is there anything like a string core or kernel?
That I don't know. That's a very good question. But how many times is the image data more than the string data? The trade-off looks like, in the Genome Sciences Center in BC, they are keeping the samples instead of the image data now. They're getting rid of them. Yeah. So I guess it's more cost-effective to just keep the sample.
I think nobody's keeping the images anymore.
Right. They used to, up until very recently. But it's a good question to ask.
Did you prove any bound on the approximation ratio of the algorithm for the core string problem?
That's a very good question. Let me see. So, yeah, there are some bounds. The cores that we obtain are approximations to the kernels that I'd like to get. But they come with several caveats. Number one, their sizes are variable. Number two, the bounds don't say how far off from minimal they are. So in terms of the size of the collection. But they prove the following. There will be very limited overlaps between these cores. So if I give you a string, there will be very few of these cores covering everything except in the margins. They are designed for infinite strings, right? They are designed for covering an infinite string. You cannot come up with kernels to cover all of finite strings. Thank you.